Good morning, everyone. My name is Pier Andrée Penttilä, and thanks for joining me today. Today I'm going to talk actually as a follow-up to Fan's great talk about flow cytometry and her very in-depth discussion about that. I'm going to focus today on some of the technical considerations in optimizing fluorescence-based cell sorting. So there are a number of different ways to sort cells, but for the purposes of today's talk, I'm going to focus on fluorescence-based, electrostatic, droplet-based sorting. So there are a couple of images in the first slide that I have here, and you're probably wondering what they all have in common. So I'm gonna try to attempt to talk about that today and join all these various disparate kind of technologies and how they formed the cell sorter. So in the background, we have a picture of a Coulter counter, which is kind of a device used in hematology to count red blood cells, which was developed by Walter Coulter. Then we have an image of an inkjet printer, and as well as a typical modern flow cytometry sorting device here with lasers. So we will quickly see what all of these three things have in common in the development of modern day cell sorters. So normally people kind of end their talk with some useful references and guides, but I like to start with that today because all of these things are very important in understanding both the history of flow cytometry, and I've also included some seminal papers here that are very useful in understanding the development of the cell sorter. We also have the International Society for the Advancement of Cytometry and Flow Cytometry UK, two very excellent resources for everything related flow cytometry. And if you're interested in kind of going down the flow cytometry rabbit hole, things like Practical Flow Cytometry by Howard Shapiro, and also the volume 10 of Cytometry by John Paul Robinson also gives a very, very good overview of the history of cytometry. So I'll just start a little bit about talking about flow cytometry. What is it? Fan already gave this overview, but I'll talk about it just briefly to start. So it's a sophisticated cell imaging technology in which a population of cells are hydrodynamically focused into a single cell stream and subsequently pulsed with lasers to record fluorescence at different wavelengths. Fluorescent dyes and probes are typically used, and this allows the analysis of the expression of a variety of different cell markers, such as proteins, for example. And fluorescence-based cell sorting, actually, in this context that I'm discussing it, uses electrostatic deflection to purify populations of interest based on these fluorescent properties. And cell sorting can generate purities of over 99%, currently making it still the most effective technology for fractionating mixed populations of cells. So to give you just a brief outline for my talk today, I will start a little bit about talking a little bit about the history of cell sorting, moving on to discussions of briefly about applications and measurable parameters. I will then discuss how exactly does cell sorting work and some of the factors that are necessary and very important in instrumentation settings. And I will finish my talk discussing a little bit about helpful guidelines and tips for sample preparation. Here we have an image of the patent that was developed by Mac Fulwiler, who's actually kind of the father of the development of a cell sorter. And this was published in patented, excuse me, in 1968. So Mac actually developed cell sorting over 50 years ago. And in 1962, he actually joined the Los Alamos laboratory in New Mexico with Marvin Van Dilla. And their primary interest at the time was actually in monitoring the fallout from atmospheric nuclear weapons testing that appeared in food products. Unfortunately, very much relevant in today's geopolitical climate. So upon signing the Atmospheric Nuclear Test Ban Treaty in 1963, his attention actually turned to how his team could assist the biologists at Los Alamos. And he became interested in the Coulter counter, which was, as I mentioned previous, developed by Walter Coulter. The Coulter counter enumerates and sized particles suspended in electrolytes. These particles are pulled through a small glass tube concurrent with an electric current and produced a change in impedance that is proportional to the volume of the particle traversing the orifice. This pulse in impedance originates from the displacement of electrolyte caused by the particle and was actually being used in hematology to count and size the various cells making up whole blood. Fulwiler noticed that a pathologist in the group at the time was actually incorrectly interpreting the existence of what he believed to be a population of immature red blood cells due to misusing the instrument. So in typical scientist versus scientist fashion, Fulwiler was then motivated to prove him wrong if he could just develop an instrument that could physically separate cells based on an electrically generated signal. He read a paper by Richard Sweet, who actually had developed an ink writing oscillograph, or what is now known as an inkjet printer, where a jet is vibrated, breaks off into drops, which were then charged and subsequently deflected to be printed on a variety of different objects. So Fulwiler immediately thought that these small droplets would be actually ideal carriers for a cell. And he coupled this idea to the Coulter counter and then patented the device in 1968. Now, the technique of fluorescent-based cell sorting was actually further expanded by Leonard Herzenberg in 1969. And this is an image of him here during the time that he did a sabbatical here at the LMB. Leonard Herzenberg, who was a geneticist at Stanford, was actually interested in the addition of a light source in order to sort fluorescently labeled cells for a more specific fractionation of certain subsets. And he actually discussed this idea with Fulwiler. Fulwiler actually decided he wasn't very interested in developing that type of device, but he actually gave Herzenberg the plans for his cell sorter, where lasers were subsequently added to the device. This was later developed by Becton Dickinson, and the term fluorescence activated cell sorting, or FACS, which is very commonly used in flow cytometry, is a trademark of theirs. And then, of course, upon the invention of the monoclonal antibody technology developed in-house here at the LMB by Cesar Milstein and George Kohler in 1975, this actually made the marriage between what we know as the marriage between the field of immunology and flow cytometry cell sorting complete. Scientists now had the ability to tag antibodies with a fluorescent dye and identify and fractionate cell populations of interest for downstream genetic screening or transplantation experiments. Now, there is a broad variety of applications used in flow cytometry across both the biomedical and natural sciences, and across very diverse fields, ranging from, as I mentioned, immunology, of course, molecular biology, pathology, medicine, and even the natural sciences like plant biology and marine biology, as well as protein engineering. It's become really a crucial tool. And some of the measurable parameters are listed here. So we have, of course, things like we can measure cell surface and intracellular antigens, protein expression or phosphoflow as we know it, total DNA content, cell cycle analysis, cell kinetics, proliferation, ploidy and aneuploidy. Of course, transgenic products, which are labeled with fluorescent proteins, for example, green fluorescent protein, apoptosis and cellular viability, so the quantification or measurement of DNA degradation, mitochondrial membrane potential or caspase activity, of course, chromosome analysis and chromosome sorting is very important. And even things like cellular pigments, chlorophyll, or phycoerythrin. and the characterization and abundance of community structure of plankton as used in marine biology. So you can see it's become, again, a very, very important tool across a diverse array of fields with many different types of measurable parameters. One of the kind of more fundamental aspects of a flow cytometer, and more specifically in the context of a cell sorter, is the aspect of the fluidics of the system. And this is very, very important in the proper functioning of a cell sorter. So the fluidics system actually consists of a central core through which a sample fluid is injected. And this is enclosed by an outer sheath fluid, typically a saline solution. And due to the narrowing of the sheath in the nozzle, for example, the fluid velocity is increased. And the sample is introduced into the center and is actually focused by the Bernoulli effect. And this allows the creation of a stream of particles to flow one cell at a time, which is very important because cell sorting and flow cytometry is a high throughput single cell analysis technology. And this is known as hydrodynamic focusing. And under optical conditions of laminar flow, there is no mixing of the central fluid stream, which contains the sample and the sheath fluid. A very important factor in terms of characterizing the stability of fluid flow is known as the Reynolds number. And we have that equation here where D is defined as the diameter of the stream in centimeters. Rho is the density of the fluid in grams per centimeter cubed. And then we have that times the average fluid velocity in centimeters per second, all divided by eta, or the fluid viscosity, which is represented in poise. And laminar flow is actually possible for values of the Reynolds number under 2300. And this is very important because any Reynolds values above 2300 can result in a very turbulent flow, which will disrupt the cell sorting process quite considerably. So these are some of the safe values of stream velocity for a range of nozzle diameters with system pressures and optimal droplet generation frequencies corresponding to those velocities and nozzle diameters. And these are calculations thanks to Ruud Hulspas, which is in Shapiro's practical flow cytometry. So you can see at a typical nozzle diameter that we use for cell sorting at 70 microns, you can have a fluid velocity, a safe fluid velocity to mitigate or minimize any turbulent flow at about 32.8 meters per second. So this all leads to, I guess, probably a very important question, which is how exactly do we perform electrostatic cell sorting? So this slide represents two different types of cell sorters that are commonly used in the field. We have on the right-hand side a jet in air system, and on the left-hand side a quartz cuvette system. First, I will more or less explain how electrostatic droplet sorting takes place, and then I will briefly discuss the differences between these two systems. So we have a conductive sheath fluid that's used, as I mentioned, typically a buffered saline. And the flow chamber is actually vibrated by a means of a piezoelectric transducer. And it can be vibrated at a frequency anywhere between typically around 20 and all the way up to upwards of 100 kilohertz. And this vibration causes the fluid emerging from the nozzle to break up into tiny droplets. The flow chamber, and hence the flow stream, is actually charged at the moment a cell of interest is inside the droplet currently being formed. And the stream of droplets passes through a pair of charged plates so that the charged droplets are deflected, of course, according to the opposite charge, and collected together with the cell contained therein. Therefore, cells in any defined gates in advance can be sorted using a positive or negative charge. Some of the fundamental differences between a jet and air and a quartz cuvette system are that the interrogation point, so the laser interrogation point, the point at which the cell passes or is interrogated by the laser, is in a jet and air system located below the nozzle point. In a quartz cuvette system, such as the BD Fax Aria, the interrogation point actually takes place above the nozzle. And what this means in a practical sense is that there's far less dead time typically. So jet and air systems. So jet and air systems tend to have a much faster rate of interrogation and can actually be run much faster than typical quartz cuvette systems. That being said, both systems are very fast and can run upwards of anywhere from, let's say, 5,000 events per second all the way up to about 50,000 events per second. This, of course, depends on the type of cell that you're sorting and what you're trying to analyze. A very important aspect in cell sorting is something that we refer to as the drop delay. And this is extremely important for two reasons. First, for the proper recovery of cells downstream, and also in making sure that the purity of the fractionated population is of the highest possible value. So since the frequency or the kilohertz in kilohertz is known, the time taken for the cell to traverse the distance L, so that is the distance between the interrogation point by the laser and also the point of breakoff before it's broken off from the stream to be collected downstream, this can be calculated. And this time determines when to actually charge the stream as the required cell breaks off into a drop. And this is known as the drop delay calculation. This is extremely important because if the drop calculation or the drop delay calculation is not properly performed, we can either get a much lower recovery of cells than what would be expected, or the purity of the fractionated population suffers as a result. There are very important factors that govern droplet formation, and the first, of course, is the transducer frequency. So as I mentioned, the frequencies can range anywhere between about 20 all the way upwards of about typically 90, but can be higher than 90, about 100 kilohertz. The amplitude of the transducer is also very important, of course, in the droplet formation. Things like the nozzle diameter, which I will discuss in a moment, are also very important in droplet formation. Sheath pressure, typical sheath pressures of a cell sorter can range anywhere as low as about 9 or 10 psi and upwards of about 70 or 80 psi. And of course, finally, the phase of the charging pulse. So now I will briefly discuss some of the factors or problems that would affect drop formation in a typical cell sorter. And the first is actually temperature. So the viscosity of the sheath fluid is actually temperature dependent, and this is why lower temperatures are always desirable, and why often, if you've ever been in a cell sorting or flow cytometry facility, you will want to put on a sweater. So we're known to keep the temperatures quite low. And that is, of course, because higher temperatures will cause and disrupt or cause disruptions to the laminar flow, which is something we want to avoid when we're sorting cells. Things like debris in the nozzle, of course, will affect drop formation. You can imagine if there is something blocking the nozzle or partially blocking the nozzle, that will disrupt proper drop formation. Anything like that's partially blocking the flow cell will also do the same. Any kind of air bubbles, either in the fluidic lines or in the flow cell itself. And the takeaway message here is that a clean or bubble-free sorter is really a happy sorter. So in flow cytometry facilities, we spend a lot of time making sure that the system is free of air and free of any debris, either in the nozzle or in the lines itself. A very common question that a lot of users ask anyone in flow cytometry is more or less, how do we estimate drop volumes generated by cell sorters? And you can calculate for any nozzle orifice as a proxy of the diameter of the drops, and thus its volume, using the calculations that are on this slide. So it is a proxy because you're assuming that the drop is a sphere, and it will never be a perfect sphere, but I think this is a pretty good estimation. So the diameter of the droplets is approximately 1.89 times the jet diameter, and you can calculate the jet diameter by multiplying 0.866 times the nozzle orifice diameter. And so if you use this calculation here, you can see that, for example, in a 70 micron nozzle, your drop volume per drop is about 0.8 nanoliters. And compared to a 100 micron nozzle where it is 2.3 nanoliters. For many people, this is particularly important for anyone trying to, for example, purify or fractionate populations of a very large number. There are occasions where they want to make sure that they minimize the amount of saline or buffer that's in the solution. In particular, for anyone doing any kind of downstream genomic applications or single cell genomic cell sorts, where they are particularly concerned with a very low droplet volume in nanoliters. One of the other factors that is very crucial in appropriate cell sorting techniques is the selection of nozzles vis-a-vis the size of the cell that or particle that you wish to fractionate. So the drop stability and recovery are actually best when the particle size is less than or equal to the nozzle diameter divided by 5. And it's usually very poor if the particle size is greater than the nozzle diameter divided by 4. So a very common misconception in flow cytometry or cell sorting is that you can, put through, if a particle is, 30 or 40 microns, you can easily put that through a 70 micron nozzle, and that's certainly not the case. So this is a general guideline in terms of the types of cells that you may encounter in cell sorting, and the appropriate nozzle size vis-a-vis the cell type. So for a 70 micron nozzle, you probably don't want to push through a cell or a particle that's larger than 14 microns as a general rule. And in a 100 micron nozzle, you probably don't want to go above 20 microns. Most modern cell sorters have quite a wide range of nozzles available that can be adapted. So for example, in our facility, we actually have systems that can use anywhere from a 70 micron, an 85, 100, 120, or 150 micron nozzle to accommodate a variety of different cell types and cell sizes or particle sizes. Now, there are very distinct types of sorting in terms of what is known as sort modes. And so the 3 different types are listed here. So the first is yield. And this is where sorting the number of recovered cells is the primary concern. And each target particle is sorted whether or not a contaminant or non-target particle is close enough to be sorted along with the target particle. The second type of sort mode, and this is probably the one that most people are particularly interested in achieving, is that of a high purity. And sorting where purity is a major concern and maximizing yield is desirable. So this is where a target particle is sorted only if no coincident particle is detected and if no known contaminant particle is included in the droplet or droplets sorted. And finally, this third major type of sort mode is known as single cell. And this is when you want to sort for maximum achievable purity, irrespective or regardless of the cost in the yield. So this is very important for single cell cloning experiments, for example, or single cell genomic experiments. And the presence of any particle, contaminant, or target in a 3-drop envelope surrounding the target particle causes the sort to be aborted. So it's extremely stringent, but at a very high cost in yield. And then in the next slide here, I will just try to explain a little bit how these decisions can be made. So if you look at the sort precision in yield mode, you, as an example, you can see on the right-hand side here, a target particle in gray and a non-target particle in red. And so in yield mode here, there are always 2 drops sorted, regardless of the contaminating cells, and no cells are lost. So you can see that if the target particle here falls within this blue area, it is sorted, and it's still sorted even if a contaminant particle is adjacent in the adjacent drop or close enough to it. So you can see that in this instance, you will get a much higher recovery of cells, but it's going to be somewhat at a cost of purity. Now, in the instance of a purity mode, if possible, 2 drops are sorted without contamination of a non-target cell in the neighboring drop. So in this instance, you can see that a non-target particle is in this red area of the window of the adjacent drop. And so in this instance, it will actually not sort this target particle because a non-target particle is too close to the drop being unsorted. So the only instance in which it does sort the particle is if the non-target particle is outside this mask or this area of the adjacent drop. And then finally, in the single cell mode, a drop is sorted if the cell is actually directly in the middle of a drop. And if a cell lies in the border, a sort abort occurs. So this is why it is very stringent and why this is at a huge cost in yield. So for example, here you can see that it will not sort this particle because there is a contaminant particle. Even though it's in the middle of the drop, there's a contaminant particle adjacent to it. So again, this is a very useful mode for things like anything that requires single cell cloning or a very stringent assurance that you only have 1 cell in a well. Now there are, on top of that, other considerations in terms of what we define as sort envelopes. And there are 5 different ways to actually sort a target. The first is 1 drop deflected only if the particle is actually centered in the drop, as in the previous example that I had for the single cell mode. 1 drop deflected wherever the particle is located in the drop. You can have 1 or 2 drops deflected, 1 if the particle is centered, and 2 if it is close enough to the edge that it might drift into the next drop. You can always sort 2 drops deflected, 1 ahead or behind, or finally always 3 drops deflected. And I will attempt to explain a little bit by using an example of how sort envelopes vis-a-vis specific types of samples are important in this type of consideration. So here I have, based on the paper by Van Dyck, a graphic representation of the theoretical interaction between, in this case, filamentous cyanobacteria in the jet and droplet formation in connection with the sorting envelope and the drop delay. So this is a very interesting example because these are long filamentous bacteria and not the typical kind of round cell particle that would kind of nicely fit inside a drop. And the purpose of this paper was to basically try to find the optimal nozzle and pressure size, as well as the drop envelopes that would be ideal in sorting these very long filamentous bacteria. So we have first two different drop delays at time 0 and at time 0.1. And the droplets at the delay 0 will envelop the whole filament, or at least the front of it, depending on the size of the droplet and the length of the filament. As you can see, the breakoff point is right here. It's the same in both instances. And if the filament is long enough to span over 2 or more droplets, droplets at delay +1 or higher will envelop the rear or the middle section, actually, of the filament. So short filaments should thus be sorted by the drop delay 0, while longer filaments actually can be sorted by potentially multiple delays. And in this paper, they actually tried using a 1-drop enriched envelope versus a 3-drop enriched envelope. And sorting 3 droplets at once rather than a single droplet, in this instance, increased the chance that the droplets successfully envelop the filament and allow it to be sorted. And that's because these filaments are particularly long, as you can see represented in this graphic. But sorting with an envelope larger than 1 droplet is actually only more efficient when it is used, of course, in combination with the most suitable tip size. And in this instance, it was actually the 100 micron nozzle tip, and the drop delay in relation with the length of the filaments that are to be sorted. So you can see that from here, cell sorting is not always so straightforward. There are very important factors to consider, particularly with not only the size of the particle being sorted, but actually its shape. So I'm now going to talk a little bit about purity and recovery and what these mean in the context of cell sorting. So in terms of recovery, not all the cells sorted into a tube will be recovered after you spin them down. And we need to consider how many of the cells of the ones we want are actually deflected into a tube as a measure of the sorting efficiency, or in this case, the % sorted of the desired population. So that's why it's incredibly important once you receive your sorted cells to spin them down and to actually do a quick count so that you are comparing the numbers that were given to you from the cell sorter to what you're actually recovering. And finally, purity is obviously of extreme importance, because what we're doing is we're making sure that we're assessing the % of the target population that was successfully in the sorted cells. So if possible, we always rerun a sorted sample to actually validate the post-sort purity. Of course, this isn't always possible in, for example, single cell sorts or very rare subsets. But if there are enough cells sorted, we always rerun that portion to validate that purity during the sort. And the verification of the purity and recovery are important. First, we assess some of these things based on the Sidestream stability. So if the side streams are tested and are stable until the sample goes on, then that typically indicates that there are problems with the sample preparation. And I will talk a little bit about how sample preparation can affect your cell sort later on. But when recovery is less than expected, it is typical or likely that the sample does not have a Poisson distribution, and that typically is because the cells are too sticky, there's too much serum, or the sample prep is not very good. So you can add things like DNAs to get rid of dead cells and to reduce that stickiness. The other aspect of improper or less recovery than expected is a choice of an inappropriate nozzle vis-a-vis the size of your cell. And this results in an unstable droplet generation. So the first thing to do is to make sure that you understand and know how big the cell is or the particle is that you want to sort, and to verify that against a bead standard first if it's unknown, as this will very much help us decide what type of nozzle to go ahead and use. Now, I will talk briefly a little bit about some sample preparation and guidelines for successful flow cytometry cell sorting. And this often requires that there's more attention that's paid to sample preparation than is typically done with just preparing samples for just running on an analyzer, for example. And of course, the challenge here lies in balancing the fractionation of your cells in a timely way with preserving their viability, their functionality, or expansion and culture for downstream assays, for example, like cytokine production. So a prerequisite is to have a good single cell suspension that ensures the best possible flow while you're minimizing doublets or two cells that are stuck together, for example. So the buffer choice is really key. And what we recommend is just DPBS or HBSS, calcium magnesium free, with about 10 to anywhere from 10 to 25 millimolars of HEPEs and a little bit of serum or BSA. What we don't recommend actually is RPMI or DMEM, and they don't make the best candidates for sample sort buffers or sort collection buffers, since first, they are a different buffer type than the cytometer's sheath fluid, so bicarbonate versus phosphate, and by design require 5% CO2 to maintain physiological pH, and usually contain divalent cations and phenol, which is very, very fluorescent and contributes to additional autofluorescence, thereby reducing a bit of your resolution. If a bicarbonate media is used, you should be wary and use either calcium or magnesium calcium or magnesium free formulas without phenol, or you can mitigate the undesirable divalent cation effects for sorting by adding 1 millimolar EDTA to 25 millimolar HEPEs and a bit of protein. Another note about HEPEs buffered bicarbonate media, it's actually reported to be light sensitive. And so it's a generally good idea to protect any sample for flow cytometry cell sorting from light. So I'm going to talk briefly also about the difference between adherent cells and cells isolated from solid tissues compared to, for example, other types of cells. There are certain considerations, special considerations for these types of cells. So the first is to ensure that you're inactivating, if you're using it, trypsin with soybean trypsin inhibitor, or use acutase. And cells grown in media with serum, your culture should actually be rinsed twice with PBS prior to dissociation. This helps viability of these cells. Make sure you test your dissociation with non-enzymatic buffers that relying on chelating agents, because this can also be quite, can affect viability quite significantly. And always remember to use the highest quality reagents for collagenase, pronase, dispase, or trypsin, because any of these things can significantly affect your sample preparation and downstream viability even prior to sorting. And finally, collagenase is also dependent on calcium for activation, so do keep that in mind. Now, a little bit about collection tubes and buffers. The first is it's best practice to pre-coat collection buffers with serum, and that aids for the cells if they are kind of, for example, hitting slightly the side of the tube to kind of fall inside into the collection media below. Polypropylene tubes are actually preferable over polystyrene tubes because the cells adhere less to the tube wall. And polystyrene tubes actually can build up the charge of the deflected drops on their surface. And this can actually result in cross-contamination between the collection tubes due to kind of jumping drops caused by the repulsion of incoming drops. You should use the same sample buffer for the collection tubes with 2 to 10% serum if desired. And any media added to the well should be HEPES buffered and conditioned beforehand. Again, do not use unbuffered bicarbonate media to the collection tubes, as the sort buffer added will increase the pH, causing undesirable salts to form, thus reducing viability. And sort into wells or Eppendorfs for very rare subsets. You can imagine that if you're sorting into a 15 ml tube, only a couple of 100 or 1,000 cells, it has a very, very long way to fall inside of that tube. And when you spin that tube down, your pellet will be extremely small. So your recovery is typically better for very rare subsets when you sort into Eppendorfs. And as I mentioned, always choose polypropylene tubes that over polystyrene for collection. So just to summarize a little bit about cell sorting, and I think the primary importance here is communication with the people that are sorting your samples. So keep in mind that it's really important for us to understand what your downstream experimental goal is. Is it culturing or enrichment? Is it a genomic experiment? Is it transplantation, any kind of functional assays? Or is it enrichment for cryo-EM? All of these different types of downstream experiments will affect the different nozzles or different types of, for example, parameters that we choose to tweak the cell sorter in order to get you the best possible downstream results. And you should always consider that cell sorting is a balance between speed and cell sensitivity and pressure. So it's not always the case that it's better to just run the cells through at the highest speed. Even if you do need a lot, there is a balance between the two. As you can imagine, pushing cells too quickly through a cell sorter can also cause mechanical stress to the cell itself, which is something that we actually would like to minimize as much as possible to ensure that you're getting a very viable product at the end. And I'm happy to take any questions. Thanks very much for your attention. Could you sort phase condensated protein droplets from bulk protein in solution? Ooh, that's a good question. That's a tough one. I've never done that. I don't think you can do that, but it's a good question. I'd have to have a think about that, Chris. I'll leave it to you to always ask the hard question. Yeah, very good question, actually, but I'd have to have a think to answer it properly. I've never done it, so, and I don't know anyone who has. So it might be possible, but it might not be. I'd have to think. Oh, good question from Hannah. So can you adjust the nozzle in every cell sorter, or is it fixed? Very good question. I probably didn't make that obvious enough in my talk. It's not fixed. You can adjust it. And it depends though on the cell sorter. So there are some cell sorters where it's actually fixed. And I will say those tend to be in the kind of the less let's say, cheaper range of cell sorters, shall we say. But the more adaptable sorters typically have a range of nozzles that you can swap and change as necessary. So the only thing to consider there is that if you are adjusting the nozzle, for example, in the same day in a jet and air sorter, it can be a little bit more difficult to swap nozzles and have stability in the stream. So for example, if I'm using a 70 micron nozzle in the morning for a particular type of cell, and then I switch to 100 in the afternoon, typically you need a few hours in between to allow after that switch for the jet and air sorter to kind of stabilize in terms of its fluidics. In a quartz cuvette sorter, such as the ARIA Fusion that we have in the facility, it's actually much easier to swap the nozzles. It's much faster. So your downtime in swapping the nozzles in between setups is much less.